looks like this. So wherever there is branch, it is present like this. And all this like all the regions where there is no branching is merged into a single node. So once a standard graph is built, then it is converted into a compacted graph, and then the algorithm will write each path into your assembly file uh, separately. So one path will be like this, and the other will be like this. So those paths are written into separated sequences, which we call unit eggs. Okay, so again, there are some challenges. So repetitive sequences again create these ambiguous branches. You can see this is an animated picture of such graph. It has so many branches, and it is very intensive task to resolve this and write every single path that is present here. Okay, so this was the debugging graph approach, and there are some more aspects. So there are linear paths, which I already explained to you guys, that what is a path, and then there is tangles and knots. So you can see these uh, so where there is multiple branches are present, you can consider that as a knot or tangle. So in this picture, you can see a single branch, but there can be multiple branches. Like in this picture, you can see it is very complicated here. So it's a very like heavy knot present somewhere here and here also. So these are called tangles or knots, which are formed because of repetitive sequences. Uh, okay, and then yeah, I already explained you guys what is a standard graph and what is a compact graph. Now we'll come down to debriefing approach versus overlap layout consensus assembly. So let me first explain what is overlap layout consensus assembly. So basically what we are doing here is we are taking each sequence and we are just finding the overlaps, what is usually what I have explained in the de novo sequence. Like here, this is what you call overlap layout consensus approach. It will just find the overlaps and will convert into contigs. Debriefing is very new, not very new, but it is considered better than overlap consensus. So we are going to discuss its advantages and disadvantages further. Okay. So let us revise what is a divergent graph approach again. So method. So first it breaks the reads into k-mers. What are k-mers? I already explained. If you guys have any doubts, you can ask me. So first it breaks the sequences into k-mers, then builds a graph. that standard graph and then compact graph, where nodes are k-mers and edges represent the overlaps. Then computational efficiency. So it is much faster than your overlap consensus approach and much more memory efficient for short reads. This is considered best for Illumina short reads, which are around 50 to 300 base pairs longer. And some advantages are there. It handles the high coverage extremely well. So in case of de novo, I have already told you in de novo approach, we need a good coverage to resolve the repetitive issues. So it handles this high coverage sequences very well. Then it is computationally scalable to large genomes. It can easily assemble your large genomes like human genome, or with the repeats which are longer than its k-mer size. So if you choose a k-mer size which is short shorter than a repetitive sequence in your genome, that will be a problem. Then is so basically you are breaking your read sequences into more shorter sequences, so it might lose the connectivity information, so we don't track which k-mers come from same original read. Okay, so examples are there of the tools which utilize this de-region graph approach. So are Spades, which we will be using for our demonstration. Then Velvet is a little bit older. Velvet and Soap de novo are little bit older tools. And then there is overlap consensus approach. This is older than divergent because it is more divergent is more better in comparison to this. It is evolved much later. Then yeah, so what method it employs is it finds all pairwise overlaps between reads, builds an overlap graph, and finds the consensus path as I have explained in that picture. Now computational efficiency, so it's computationally very intensive. It has to find like it has to check for each sequence which pair will be overlapping with it. So for one, it has to uh, check with every sequence present in your data. So you can like think of how much how much possibilities are there for a single sequence to find a partner in that large number of genome sequences. Sorry, read sequences. Then it though it is not good, that much good for shorter sequences, for longer reads that comes from PacBio and Nanopore, it is very good. Then advantages are there. It preserves the read conductivity. It better handles the longer reads or longer repeats, and it produces longer context. So your assembly will be more contagious and not much fragmented in comparison to de novo. Then disadvantages are there. It is slower on larger data sets. Then it is also memory intensive, and it's also sensitive to sequencing errors. The examples of tools which utilize this approach are Kanu and Miniasm and Fly. So there are some modern hybrid assemblers also which combine both the approaches and leverages the advantages of both the approaches. so uh, now we'll take the lunch break and we'll join again and we'll discuss more about the assembly methods and what is coverage and what is read depth. Then we will discuss the key matrices of the assembly.